Everyone, please sit down. Okay, today I'm going to talk about lymphocytes. Are they friends or foe? It's about tolerance, and I'll be discussing the immunological tolerance of T and B cells and its significance, then the T regulatory cells formation during tolerance and its role in preventing autoimmunity. So how T regulatory cells can prevent autoimmune diseases, the mechanism how microbes are involved in autoimmune diseases, and finally the genetic and environmental factors involvement in autoimmunity. So we'll be discussing these four aspects in detail. The first thing, what you have to remember is that our immune system creates a lot of B and T cells, especially their receptors. That diversity has to be created randomly. That's one thing. And because it's random, so we don't know whether our T cells and B cells, they will attack your own body system or they will attack the pathogens. So this is something which is very difficult and it's against each other. So here you can see in red, 10 raised to the power 15 is the number of possible random receptors that are produced by T and B cells. And they can be your own body structures or they can be of the pathogen. So that's a difficult challenge. That's a task that body has to meet with. Now, if you see all these body organs, whether it is kidney, spleen, liver, intestine, anything, it is made up of proteins. And on the other hand, we have got pathogens, bacteria, viruses, parasites. They are also made up of proteins. So now the challenge is the body organ proteins, which are termed as self proteins, or the pathogen proteins, which are termed as foreign antigens. How your immune system differentiate between the two. So I'll just put here some scenario for you. So you have got three organs, for example, a liver, a kidney and lung. They have got protein structure, and remember, one T cell at a time recognizes only one antigen, one protein. So this T cell will not recognize kidney. This T cell will not recognize liver. So I am, for simplicity, putting just one cell. There are just a few cells that will recognize one thing at a time. On the other hand, if we think about bacteria, so recently I have taken TBL of TB. So let's say a TB pathogen, then Streptococcus pneumoniae. Now these T cells will just recognize these two proteins. So this has to be very clear. Now these T cells which recognize your own body organs, they are called as autoreactive. Okay, you will remember this word. These are called as autoreactive. And these proteins are called as self antigens. These proteins are called as what? Foreign antigens. And these cells which will damage them are called as effector cells. So now are you clear about some terms? That the T cell that recognizes your own body protein will be termed as autoreactive cell, autoreactive T, autoreactive B. The antigen will be called as self, and the effector T cell is against the pathogens. So they will destroy them. Now ideally, what should we do to protect ourselves from autoreactive T cells? What should you do? Kill them. Best thing is to kill them. Delete them from your body. But sometimes we will see how this deletion occurs, but sometimes some of them still stay in the body, so there should be another cell to counter them or to control them, and those are called as regulatory T cells. So here is this cell, regulatory T cell. If let's say this has been deleted, this has been deleted, just one of them is remaining, this will come and block it. So for simplicity, now are we clear about all the main terms? So what is self-antigen? What are autoreactive T cells? What are effector cells? The bad cells, autoreactive cells, have to be deleted, or they have to be blocked by regulatory T cells. So now this is the challenge that we see. The immune system has to recognize between self and foreign antigen, and this challenge is met by the process of tolerance. So if you read this definition of tolerance, tolerance can be defined as state of unresponsiveness to self-antigen. So you don't want your body to be attacked by autoreactive T cells, so either you kill them or you block them. This is called as tolerance. Now where this deletion will take place, how you can get rid of these autoreactive T cells, you have to do it in the thymus. So T cells, they are produced in the bone marrow, and then where they are matured? Thymus. That maturity is basically filtering or deleting the bad cells and producing the regulatory cells. So thymus, you remember I told you, I'll tell you in detail what will happen in the thymus. So this is today's task, that we will find out that after going from the bone marrow into the thymus, these autoreactive T cells, possible autoreactive T cells, they will be deleted, and some new regulatory T cells will be produced to help them in this process. So when it is in the thymus and for B cells in the bone marrow, this is called as central tolerance. So this, you can see the figure of thymus, and you can see inside the T cells are deleted, deletion is taking place. And also in the bone marrow, the B cells will be, bad B cells will be deleted. That is the maturation in the bone marrow. 95% of the T cells that come from the bone marrow, they are deleted. So it's a very harsh process. So imagine if 100 cells are produced, 95 will be destroyed. Only 5 will remain that will do the function of immunity. And still some of them will not be good. So out of those 5 which are remaining from the 95, they might be, one of them might be a bad autoreactive T cell. So to control that, you can see here, this green cell is actually the regulatory T cell. And on the other hand, B cells come out directly from the bone marrow. They don't have to go to the thymus. And now the autoreactive cells, whether possible autoreactive B or possible autoreactive T, they will be suppressed by a regulatory T cell. So you can see he has a regulatory role on B cell and T cell. Now we'll see what happens in the thymus. So this is, just consider it as thymus. It has got 2 portions. One is the outer portion that is called as cortex. From the bone marrow, T cells come. These are called as, which T cells are these? Immature. These immature T cells will enter here in the cortex. When they are immature, so they don't have any receptors on them. What are the possible receptors they should have? They can have CD4, CD8, TCR. So first cell that enters, it has no receptors. That's why, because of CD4 and CD8, it is called as double negative. Okay, so it has entered, neither it has CD4 nor it has CD8, so it is called as double negative. After just a little time, they will express both CD4 and CD8. So now they have both the receptors, CD4 and CD8. Now this stage is called as double positive. Okay. Now there is another cell in the cortex. So its name comes from the cortex. It is called as cortical thymic epithelial cell. Okay, cortical thymic epithelial cell. So there is, I'll just draw 3, so all of them we take into consideration. These cells will express random proteins, but important point is it will be with the help of MHC class 1 or MHC class 2. So let's say here is MHC class 2, here is MHC class 1, and in the middle there are possibilities of MHC class 1 or 2, but there is slight difference. So they entered as double negative, they expressed all CD4, CD8, TCR, these are called as double positives. Now either they will interact with MHC class 2 or they will interact with MHC class 1, or the protein will not be recognized by the T cell. So these are the 3 possible scenarios. One, it is recognized through MHC1, one recognized through MHC2, one is not recognized. So the ones which are recognized, that will convert into single positive. So if it recognizes MHC class 2, so which cell it should be? So the one that comes into the cortex now here, this is T cell with CD4, and this cell which comes here, T cell with CD8. These are single positive. Okay, and this cell which is not able to recognize the protein, it will die of neglect because it was ignored. And what is special about it, when they interact, they find the protein and the MHC class 1, this cortical cell gives it survival signal. They will give it certain proteins just for sake of simplicity. They help them to live. When they interact, it means you can live. They interact, it means you can live. When no interaction, no protein recognized by the cell, it has to be deleted. Okay, so now what you are doing, you are choosing all the population. So the T cells, immature cell come, double negative, double positive, CD4, CD8, not recognized, this is deleted, these are selected. This selection is called as positive selection. So far, everyone is okay with this? Now the next portion is in the medulla. So I'll just extend. This process will take around 4 to 5 days. Okay, now what you have got single positive CD4 and single positive CD8. Here also is a special cell, and this is called as M, medullary thymic epithelial cell. Okay, this one is called as medullary thymic epithelial cell, and it has one very special quality. It has got one gene, and its name is AIR. This gene is called as autoimmune regulator. What is autoimmune regulator? You saw all the organs, liver, spleen, kidney, pancreas, they have to produce their protein wherever they are. This gene, what it can do, it can produce most of those proteins inside the thymus. So you understand, this cell, it will do ectopic expression. So what expression? That is supposed to be, for example, a liver protein, but this is capable of producing that liver protein in the thymus. Same is for liver, pancreas, kidney, skin. Every protein will be expressed by this medullary thymic epithelial cell, and one by one, any T cell which, for example, recognizes, for example, we'll now take it as one example. Let's say in the start I showed you autoreactive T cell which could recognize the kidney. So if this T cell, it recognizes the kidney, this is the autoreactive T cell, and when it comes here, it will express the kidney protein. The T cell will come. If they bind to it, if they recognize it, this medullary thymic epithelial cell will delete it. Okay, same as, let's say this CD8, it recognizes the spleen or liver. So this liver was recognized here by this T cell. When it comes and interact with thymic epithelial cell, it will also be deleted. So all the autoreactive cells, whichever recognize kidney, spleen, pancreas, one by one, they will come. They will see that if it interacts, they bind to each other, they recognize it, they will be killed, they will be deleted. That's why this part is called as negative selection. So now I would like you to think of this whole process as when you want to recruit someone, for a job, for a position, what you do? You first advertise the position and tell them everyone to come with their CVs. So in the shortlisting procedure, what you do? Qualified, keep it. Qualified, keep it. This does not belong. He is maybe a mechanical engineer, we need doctors. So just take it out. So first, you positively select everyone, all who is possible candidate. So this has been done in the first phase. And those who you don't need, you are ignoring them. So that's the positive selection. After that, now you do screening, careful screening. Is he a good candidate? He has got very good GPA. Is he dangerous for us? Should we keep him? So then you negatively select one by one. No, this is not a suitable candidate. He is dangerous. Take him out. This is one. So finally what you get is just the T cells and the B cells that will not interact with any, the autoantigens or the self-antigens. So the first phase was positive selection. It took 4 to 5 days. Then we come to medulla. It's the second phase. It takes also about 4 or 5 days. And here the medullary thymic epithelial cells express this gene, which will help it express all the possible proteins in your body. And one by one, all the autoreactive cells will be deleted. That last phase is called as negative selection. And what comes out now? You have single positive, non-reactive. So single positive, naive T cell, whether it is, it can recognize any pathogen, but it has to be single positive, CD4 or CD8. So now let's see what's in this figure. Here you can see this part which is happening in the thymus is called as central tolerance. So number one, you see they entered as double negative and then they become double positive. So this is the first stage. Now on the other hand, the cell down here, this is expressing MHC class 2. This is thymic epithelial cell. This is thymic epithelial cell. The one which will interact with MHC class 2 is CD4. The one with MHC class 1 is CD8. So this is positive selection, while some cells, they will not interact at all. So they will be ignored and they will die of neglect. No survival signal, so they will die. Now further, this same thing goes into the medulla as showed you here. Here you have got another type of special cells which are called as medullary thymic epithelial cells. They express different proteins of the body, and all the autoreactive CD4 positive or CD8 will be destroyed. Now one more thing appears here, dendritic cell. Normally dendritic cells are supposed to present the antigen, isn't it? In the thymus they will also take some proteins from this medullary thymic epithelial cell. So it will express that protein, and dendritic cell will just capture it, and then it will do the same job like medullary thymic epithelial cell. If it recognizes the autoreactive cell, it will destroy them. So you can see those who are recognized, they are destroyed. Those who are not recognized, they go free. So CD4 cells go out, CD8 cell go out. During this whole process, 95% of the cells coming to the thymus are destroyed. Only 5% will go out. Now you may argue that is this 100% foolproof mechanism? There is no chances that any autoreactive T cell don't go out? No, there is a possibility still that some autoreactive cells go out into the bloodstream. For that you have got another mechanism. So now what you have, they are mature, naive, single positive T cells that leave the thymus. Only 5%. Yes, please.
What's the role of dendritic cell?
Dendritic cell helps the medullary thymic epithelial cell. So, all the job cannot be done by this. He expressed the protein. Some dendritic cells will take that protein, which is of maybe kidney, of spleen, and then present it to the possible CD4 or CD8 cell. Whichever recognize, it will destroy it. So they do the same job, but this dendritic cell don't have AIR gene. They will take it from the medullary thymic epithelial cell. Now, just to recall, you found the immature cells which were in the bone, they came out. Then these are of course naive because they have never encountered any antigen. They enter the thymus and become mature. So now when they leave out, they become mature, naive lymphocytes. Then if they encounter self-antigen, they are autoreactive. If they encounter foreign antigens, these are effector cells. And finally, the effector cells, before becoming effector cells, they have to proliferate, differentiate, and do the selection. So I just wanted you to recall all the terms, and I hope you understand which definition belongs where. Now, this is the possible scenario that in the thymus you saw negative selection, the last phase, and the T cells, they come out. Some may be autoreactive, very few, just it's an assumption, it's an exception. So to control them, the thymus is also producing regulatory cells. So now we have to see that what these regulatory cells can do. Here in this second part, what you see is that up mature lymphocyte that has come out, it is a possible autoreactive cell, and down is what can happen to this autoreactive cell. Number one, you see the word energy. What is energy? Normally, for a T cell to become activated, what are the 3 steps that are needed? Any T cell to become active. Priming with the help of MHC class 2, CD4, TCR, then co-stimulatory signals is the second, and third is production of cytokines. So in energy, dendritic cell are not presenting the kidney antigen or liver antigen, so there is no antigen presenting cell involved. It's just the kidney cell is somewhere there and the autoreactive T cell finds it. So in the absence of priming, in the absence of co-stimulatory signal, the T cell will be switched off. This is a special mechanism that if there is no primary signal, if there is no secondary co-stimulatory signal, the T cell will be switched off. This kind of cell, which is now not dead, it's just switched off, it is no more active, this is called as anergy. A-N-E-R-G-Y. Anergy is switched off T cell. Simply, it's not functional. Number 2 is there is a possibility when it recognizes, it is killed, programmed cell death. So you can see the number 2 is apoptosis. And last thing that you see is regulatory T cell that was produced in the thymus, it's going to block these autoreactive cells. So far, everyone is okay or no? Yes, please. We'll see. There is a slide about how regulatory T cells will suppress the autoreactive T cells. So there are 4 different steps. We'll see that. So now you have moved from the thymus and from the bone marrow into the periphery. So there was one part of central tolerance, we have discussed it for the T cells, and there is now one part in the periphery that is called as peripheral tolerance. But before doing that, these are the possible three outcomes whenever an antigen comes in contact with a lymphocyte. First, immunogenic. When in a normal immune response, of course it has to be presented by an antigen presenting cell, a dendritic cell, a macrophage, or of course a B cell. When they present it, as I already said, primary signal, co-stimulatory signal, and cytokine. They all have to play a role to create an immune response. So the first scenario is they proliferate, they differentiate, and effector function is done. So this is one. Number two is tolerogenic, the tolerance that we saw here. If in the periphery, now the kidney is there, kidney cell is there, and the lymphocyte come in contact, as I already mentioned, because no primary signal, no co-stimulatory signal, no cytokine production, so either it will be switched off, which is called as anergy, or it will be apoptosed, it will be destroyed. And the last situation is that the lymphocyte cannot find the protein. And how is it possible? So there are certain immune privileged sites where lymphocytes cannot go and circulate. Can you remember any? Sorry? Central nervous system. So the myelin or brain and meninges, T lymphocytes cannot go there. So if possibly there was an autoreactive cell that can recognize that should recognize or attack a meninge, it cannot go there. It does not have the luxury to go there. So in that case, when does not see, no response. Now, dendritic cell, what is another importance of the pattern recognition receptors is that normally a dendritic cell see a pathogen with the help of PRR. That is the danger signal. The dendritic cell will express lot of things, co-stimulatory signal, CD8, MHC class 2, CD40, and if you see it is written HI, high, their levels are very high. So such a dendritic cell which recognize a pathogen and express all these primary signals, co-stimulatory signal, this will result in pro-inflammatory cytokines and it will result in an immune response. But if a dendritic cell does not see a pathogen with the help of PRR, which of course a kidney or a liver or a spleen protein, it will not be a danger signal. So what will happen? It will have all these co-stimulatory signals at low level. You can see the difference in their shapes as well. One is more active, the other one is low, it does not have lot of expression of proteins, and as a result it will result in only anergy. So does this make sense? That how tolerance in the center, you deleted the bad cells, some regulatory cells came out. Now in the periphery, if an autoreactive cell comes, it will not be presented by a dendritic cell normally. And why is that? Because there is no PRR, there is no primary signal is there, no co-stimulatory signaling is there, and there is of course difference. When no primary signal, there is no CD86, CD40, so main signals are not expressed in high quantity. Now another part that we so far not discussed is the regulatory T cell. It comes out and it can be natural. So naturally born in the thymus that comes out is called as natural regulatory cell. Excuse me. Then there are certain which are produced later on in the bloodstream. They are called as induced. So you can see here the first cell is CD4, CD8 negative, double negative cell in the thymus. They become double positive. Then they become single positive CD4, as I showed you here. And finally, some of these CD4 will express FOXP3, which is this characteristic feature of regulatory cell, and it comes out as natural regulatory cell. This above is the natural regulatory cell. While the other CD4, which comes out as single positive, they can become anything. They can become TH1, TH2, TH17, and also regulatory T cells. So if you think about what is the source of regulatory T cells, it can naturally come out of the thymus, or later like TH1 polarization, TH2 polarization, TH17 induced T regulatory cells can be produced as well. So these are the two sources of regulatory T cells. Now, as you know, he asked